Thank you, Tim, and thank you, MSTC organizers, for this opportunity. I am Bharath Takulapalli, founder and CEO of INanaBio. We are based out of Menlo Park, and also our headquarters is in Scottsdale, Arizona. I do not believe that we as a scientific community, I do not believe that we should get 90% of diseases today. We can preempt them early on. Disease preemption implies detecting in early pre-disease stages, intervening early, and preempting, curing before full disease onset. However, today technologies are not available to extract system-scale information from biology to achieve this. To contribute to this ultimate aim, we are developing two different platforms at INanaBio at the interface of nanotechnology, biology, and machine learning to extract 100 to 1,000 times more information from biological systems per every dollar invested so as to preempt disease. And I'm going to speak about one of the lead platforms that we're working on, which is a novel nanopore transistor device. It's a transistor with a nanopore at the center that acts as a transistor sensor to read out the DNA sequence at 100 to 1,000 times faster than current state of art, which is Illumina, for folks who are aware, a $50 billion conglomerate. They have about 75% of market share. And Oxford Nanopore, who is that just had IPO last year. So we are developing this nanopore transistor device with support from DARPA, thankfully, to read out the genome, and also, that's one of the first applications, also proteome and so on and so forth, epigenome for folks who are familiar. So before I dive into the technology, let me kind of inform and kind of go through why this is important. What's the use case really? Why do we need this? So what are the three grand challenges in healthcare? We want to preempt future outbreak from becoming a pandemic, democratize precision medicine, that is right drug for the right patient, and detect cancer and other diseases early on, as I was saying, preempt diseases. Preempting future pandemics or preempting future outbreaks from becoming pandemics. So as we are all aware today, current PCR tests can detect few variants of a single pathogen like COVID. That's it, at $20 to $30 cost. However, to be able to really address this challenge, we would need a single test that costs similarly $20 that can detect any number of pathogens, hundreds of viruses, bacteria, and so on, and any number of their variants at the point of care that can be scaled to populations scale screening. Precision medicine is projected to be an $80 billion industry, which is based on detecting DNA biomarkers and matching it with specific drugs to have high therapeutic efficacy. And you might have heard about Grail, Guardant, and few other companies, Exact Biosciences, working on liquid biopsy-based tests or simple blood tests to detect tens to 50, hundreds of cancers early on in stages 1 and 2. Again, they are still in development, but that's where everything is going. So what's common to all these things? The thing that's common to all these is the ability to sequence the DNA or read out the DNA bases. For a single solution to be possible for all these challenges, we need a DNA sequencer that would be robust, okay, drop-proof, solid-state based, that would be affordable. So the whole system cost should be like an iPhone, $1,000 to $2,000, versus today's $100,000 or so sequencing systems. Widely deployable for point-of-care, point-of-need testing, for military use on the field. Routine, where a 14-year-old can perform a diagnostic test for $20 per test, complex viral bacterial detection testing. And scalable, very, very important, scalable. We should be able to scale to billion tests per month and higher if needed. This is a kind of give you an idea of the growth in genome sequencing volumes, and this confirms the previous discussion on various markets and the market need. So this is an ARC Invest slide on genome sequencing volumes. So over the last few decades, the volume of genome sequence has been growing by about 10x every 5 years. And it's actually projected to grow at higher rates, exponential rates in the next decade, owing to new and upcoming clinical sequencing market, which was not so active all these days. On the right, you see about just a few of the markets which are projected to be in infectious disease cases about 65 billion by the end of this decade. And in clinical oncology, that's a 150 billion market. So totally, this whole area is bound to be or projected to be about 200 billion or higher. These are the current, as a quick overview, just to give you a state of what's today, these are the current sequencing systems that are refrigerator-sized. Illumina is the leader, 75% market share, as I said. What they do is, current technology do is, human genome is 6 billion base pairs. They chop them into smaller fragments, sequence them, and align them back to get to the full sequence. This adds complexity, very labor-intensive, cost, time-consuming. So sequencing a single genome from a single cell takes few hours to days right now, depending on the investment that you made in millions versus hundreds of Ks. So for clinical sequencing perspective or clinical diagnostics perspective, this is nowhere close to what we want to be, where we want it to be. And importantly, we cannot use these devices at the point of scale as a low-cost device for testing, population scale screening. So nanopore sequencing is the next generation of sequencing technologies, DNA sequencing technologies. So the idea of nanopore sequencing is you have a hole or a physical hole in a substrate, and you pass the DNA through the hole as you thread through a needle hole. And since DNA is a linear chain of information, if you can embed a sensor at the nanopore, around the nanopore, you can read out the DNA-based code as it passes, as the strand passes through the nanopore aperture. And it just so happens that DNA natively passes through nanopore at speeds of million bases per second. That's one whole megabase in 1 second. So whole human genome, in principle, if you have such a sensor made, can be sequenced in literally under a minute. That is probably 1,000 to million times faster than what's possible today. Right? And another aspect of nanopore technology is, since they are nanoscale devices, you could fit millions of devices on a centimeter square chip, fabricated and into a disposable chip for diagnostic tests. So very, very powerful as this thing. The leader in this area is Oxford Nanopore from UK. They use protein nanopores. So there are two kinds of nanopore platforms, proteins and solid-state nanopores. Pretty much majority of them are based on what is called ion current sensing. What it means is, as the DNA passes through the nanopore, the DNA bases have a small differential in sizes. So they block the nanopore aperture differentially. And the ions passing through that nanopore aperture are measured as a change in ion current. And by monitoring that current, you back calculate the DNA-based code. Hope that makes sense. And Oxford Nanopore has commercialized it successfully. They entered the market with products in the last couple of years. So what it comprises is a lipid bilayer. This protein nanopore platform comprises a lipid bilayer like a synthetic membrane that mimics a cell lipid bilayer, very complex. And then they perch these protein pores on top. One of the disadvantages of this approach of using protein pores and ion current sensing is, while the DNA passes through the nanopore at speeds of million bases per second, ions themselves are sluggish carriers of information. Ion mobility is very slow. Because of that, they are forced to slow down the DNA as it passes through nanopore to below 1,000 bases per second. So they're actually losing one of the major advantages of nanopore sequencing. And also, when COVID happened, they were very well commercialized, raised a lot of money in market, but they couldn't respond. That's because this platform and these protein-based platforms are very complex to scale. And manufacture billions of chips, no way. Maybe 10,000 or 100,000 chips in a few years. And another aspect of it is because they're proteins, and to folks who are familiar with proteins, they're very unstable molecules. And they are prone to biofouling, shock damage, vibration damage in shipment, and so on and so forth. So we do not yet have a solution to a point of need sequencing system that's infinitely scalable, like we do in our CMOS chip technologies. And that's kind of what we're working on. So while everyone who is working on this technology slow down their DNA to be able to read it from million bases per second to 1,000 bases, we went to NIH and DARPA and said, okay, that's one way to solve it. But another way is, why don't you build a faster sensor? And the analogy to this is, we cannot see a bullet fired from the gun, but we can see it, capture it using a digital DSLR camera. So the information is there, it's just that we need a faster sensor. So what we are developing is a CMOS transistor with a nanopore at the center, or a transistor surrounding a nanopore in 3D. You'll see that. And the transistor operates at greater than 100 megahertz. So as the DNA passes through the pore at million bases per second, it's equivalent to megahertz signal. And the transistor operating at 100 megahertz would be able to sample each DNA base at 100 times or so. So that's the idea. Then you do not need to slow it down, and by analyzing the data, you can read out the DNA very fast. Here is the technology we are building, device we are building, that combines nanopores with silicon CMOS technology. We are funded under DARPA ECHO. So we had, because this is based on silicon and we needed to build this technology from ground up, we had been trying to raise capital, and DARPA found this really interesting and promising, funded us before a prototype. So that was in 2019, and we are going to talk about a prototype we made in the last 3 years. Can we go to the next one? So there's an animation. Okay, it starts off. It's an animation we created to explain the concept. The schematic of a chip, our FENT chip, comprising an array of these FENT devices, transistor devices with holes at the center. These are 3D transistor devices. So the electron current flows from the top to the bottom, funneling around a nanopore at the center. So you take a linear transistor, make it vertical, convolute it in 3D around a nanopore. So as the electron current passes through, the DNA goes through and electrostatically modulates the electron inversion current, which has been established using the buried gate. I hope that was helpful, and that makes sense. So before we even started on FENT or even conceived FENT, we made nanowire transistor sensors that are linear and when operated in fully depleted mode, we demonstrated that we can detect small molecules with ultra-high sensitivity and specificity. This is published. So we could detect pyridine molecule, which is a benzene with one carbon, one of the carbons replaced with nitrogen. You could detect that smaller molecule with automolar sensitivity and high specificity, as you can see. So this got us thinking, the transistor has high operational speed and super sensitive. And by the way, pyridine is smaller than each DNA base. So it's almost half the size. So that got us thinking, and that's what gave rise to the FENT structure that I described. So what we have done is we have taken a linear transistor, bent it at the center, made it vertical, convoluted in 3D around a nanopore. So now the source is on the top. It goes around the inversion current that we established with buried gate, funnels around the pore and then goes to the drain at the bottom. So it's a cylindrical inversion current. And the nanopore acts as a pinch point. Here is our device prototypes. On the left, you see the device that we fabricated with our foundry, CMOS foundry partner in US. The FENT fabrication process flow is about 80 steps or higher. So deposit oxide is one step, as you know. The whole wafer is FENT devices. It's not a multi-project wafer. On the right, you see a 11 millimeter cross 11 millimeter chip with a 50 nanometer pore. That's what we have today. In future, in the next 1 year or so, we are going to combine this with the 2 nanometer pore that you have seen in the previous slide. That's work that's in progress. And we make an opening at the backside, as you can see, to access the FENT nanopores that we have built on the top side. Hope that makes sense. We have tested the frequency, operational frequency of FENT, and have demonstrated that it performs at greater than 100 megahertz with less than 3 dBm loss. When we speak of ions that other platforms, other nanopore platforms use ion current sensing, the mobility of ions in solution is about 10 to the power minus 3 centimeters square per volt second. And mobility of electrons in silicon is of the order of 10 to the power plus 3. That is, electrons in silicon are a million times faster than ions in solution. So FENT, as far as we know, is the fastest nanopore sensor to date that is faster by other sensors probably by about 1,000 times. One of the advantages we have is we can really make chips that are disposable, right? The FENT device itself is less than 2 micron. The unit cell with the reed circuit that we are now working on is estimated to be less than 50 micron. So that means we can make a disposable FENT chip for diagnostic testing that is about 2 to 5 millimeter on side, that's fully disposable, that comprises about 1,000 to 10,000 pores or FENT array that can sequence few genomes in few minutes. So which would be super powerful. You need to do that today, you need a room full of machines. That's how drastic it is. And we could do it on a cell phone sized device once it's fully developed. And this would allow us to scale manufacturing to billions of tests per month. Here is the IV characteristics of the FENT device with the nanopore integrated in silicon. So this, even with silicon, it performs as a perfect textbook transistor with off current at picoamps, on current in tens of microamps, so on to off ratios of greater than 10 to the power of 6 million. And subthreshold swing, again, is about 100 millivolt per decade. So we got lucky in getting this far. So if you see on the left side, there's 10 different devices across a whole 8-inch wafer. So that shows really high fidelity of manufacturing. We have started testing this with DNA. We paused a really long strand DNA with this 50 nanometer pore. Again, that's nowhere close to where we want to be to sequence DNA, but we started experiment anyway, just pass through and see if the inversion current responds, and it does respond. So as the DNA passes through, it electrostatically modulates the inversion current, which we can detect by monitoring the transistor current. And we have done this, again, we have done this at 250,000 acquisitions per second, samplings per second, only because the data acquisition system we were using was restricted. In future, we are going for 10 million and up samplings per second. So there you see about 50 points measured in 200 microseconds. And going forward, we'll have more. So the whole idea is we can take the data, perform machine learning algorithms and using a training data, and then in real time, quasi-real time, process the data into bins, AGCT, and what have you, and epigenetic markers, ultra fast, and then dispose of the data, replace with new data, FIFO, in very fast architecture. Hope that makes sense. The application space is huge in multi-omic analysis. not only DNA, but epigenetic sequencing, which is what our DARPA contract was. So DARPA figured out, again with others in this thing, that to diagnose disease early on, pre-symptomatically, we could do that because when we are exposed to any new, let's say, toxic or a new environmental factor, our system, our immune system interacts with that, and there's a response. And that response is encoded not in the DNA changes, mutations, but as epigenetic markers on top of the DNA. And if you can read those out, that is, read the epigenetic signatures out of immune cells, you can indeed detect disease within few days of, let's say, infection, even infection, and cancers years before they're really cancers or symptoms appear. So the science is already there. So other applications include point-of-care diagnostics, which I spoke, and at-home testing, which is my pet peeve, kind of. Even if it is 10 years, we want to make a device that you'll see a concept of, want to get it in every home. And another area that you might be following is DNA memory. So you can encode a lot of information in DNA. I'll quickly go over it. So the idea is we are developing this device to rapidly extract 100 to 1,000x more data for every dollar invested using FENT. We aim to go to research market first, most likely with partnerships. We are in touch with 5 to 6 sequencing companies. One model that we are considering is Intel Inside model, where we actually provide these chips to others so they could build a sequencer around it. But our core idea is to develop a at-home or sequencer in every clinic, and then at-home sequencer eventually. So you could do flu detection and what have you at the convenience of home for $20 using a disposable cartridge that you have on the left side. So DNA data storage, I was saying, you could store 1,000,000 times more DNA than latest memory devices. Not maybe the ones in the lab, but commercial ones. So exabyte scale data can be stored in tube full of DNA and can be stable for thousands of years. Once FENT is developed, this would really make it possible to read out that information at affordable costs. Anyway. So BOP analysis on what the FENT metrics would look like, wafer metrics. This is for 12-inch wafers. We expect about 500,000 wafers per year. That's not including at-home testing and DNA data storage markets. And if you go for 8-inch wafers, that would double up, obviously. This is my team, advisory board and board. And these are the things that we are open to partnering with, having come this far. Thank you so much for the opportunity. Thank you. Thank you all.